It's my pleasure to introduce Dr. Ed Reznik, who's going to be talking to us from Memorial Sloan Kettering Cancer Center about his work on mitochondria in tumors and the metabolic impacts there. And I guess as he calls it, the metabolic no man's land. So with that, I'd like to give it over to Ed, and hope you all enjoy the talk. And there'll be questions at the end.
All right, well, thanks for the introduction. Thanks for the invitation. And I hope you guys can hear the wonderful traffic noise in the background that's about to be coming. It's a special time right now in New York as people try and get on the Queensboro Bridge. So soon you will hear mad honking, and welcome to where I live. So yeah, the title of my talk is Travels in a Metabolic No Man's Land. And I'm a computational biologist who can't manage the slides. I'm a computational biologist, and our lab studies tumor metabolism here at Memorial Sloan Kettering. And we think about three big questions in our lab, and I'll try to cover two of them in this talk. So we think a lot about how somatic genetics shapes the metabolism of tumors. And related to that, we like to think about how phenotypes in tumors depend on the interaction of multiple different alleles of the same gene. And that comes both from our interest in mitochondrial DNA, but also in the nuclear genome. And finally, we also think about how the tumor microenvironment and its cellular composition and different cell-specific phenotypes in the microenvironment can remodel metabolism in the tumor, and specifically what the culprit cell populations are that are driving metabolic phenotypes. So I'll mostly focus on the first and last question in this talk. So I want to bring you back to what I was thinking about when I first started as a postdoc here at Sloan Kettering. And this was a time when I learned a bit about the genetics of kidney cancer. So one of the remarkable things about kidney cancer is that there are many different subtypes of it, many of which are quite rare. In fact, like the dominant subtype clear cell, I think constitutes something like 70% of all kidney cancer cases. And to each one of these subtypes, you can essentially ascribe an early truncal mutation in either a metabolic enzyme or a metabolic regulator. So for example, biallelic loss of FH or SDH causes, those are the pathognomonic genetic events in certain types of kidney cancer. Certain types of kidney cancer are associated with mtDNA mutations. And the most common form of kidney cancer, clear cell, is actually associated with loss of VHL, which is a regulator of HIF1 alpha and HIF2 alpha. So metabolic genes ultimately are really like truncal drivers of nearly all subtypes of kidney cancer. And so I got interested in studying the metabolism of kidney cancer. And to do so, because I'm a computational biologist, I wanted to understand the kinds of data that were available to make metabolic measurements. And you can summarize these kinds of data. You can put them in three bins, essentially. So the metabolic network is basically composed, it's like a bipartite network of interactions between enzymes and the metabolites which they consume and produce. So when you make measurements, you can basically make three qualitatively different kinds of measurements. You can measure enzyme levels, either with RNA-seq or with proteomics data. And we have the most data about stuff like that. You can also measure metabolite pool sizes, essentially with mass spec or NMR. And we have some data for that, a lot less than we do on enzyme levels. Or you can measure metabolic flux, so the rate of a reaction. And that is really what we are typically what we're most biologically interested in, and what's most therapeutically important. So if you give a metabolic inhibitor, it affects an enzyme, but what it's really doing mechanistically is it's usually inhibiting the rate of a metabolic reaction. So in a way, my lab's research lives, and here it comes, in a sort of no man's land, where the data modalities for which we're really data rich, which include genomics and RNA-seq, some metabolomics, are really disconnected from the phenotypes that we're actually interested in, which are rates of metabolic reactions or fluxes. And so the question that I've been grappling with, I would say, for 10 years and maybe even longer, is what we can learn about metabolism and metabolic phenotypes from indirect measurements of DNA, RNA, and metabolite levels. So it all started for me in 2013 when I was a postdoc here at MSK, and I met this wonderful surgeon pictured on the right, Ari Hakimi, and he told me about a wonderful data set that he was generating where he collected 140 clear cell renal cell carcinomas. These are the most common kind of kidney cancer, and adjacent matched normal tissue, and measured the abundance of a few hundred metabolites in each one of those samples. So up to that point, there was really no understanding of how, for example, loss of VHL in clear cell actually affects metabolism. And so we set out to define what we call the metabolomic landscape of ccRCC. And I don't want to walk you through the results in much detail, but we saw some interesting phenomena. Clear cell tumors accumulate sugars, both complex and basic. They're typically depleted of amino acids, with the exception of a few special ones. And most importantly, we noticed that the metabolism of clear cell tumors was strongly associated with the stage or the grade of the disease. And something like, I think, 15 to 20% of the metabolome was clearly correlated to the stage of the disease, the clinical stage. And what that suggested to us, or what it implied, is that changes in metabolite abundance were really hallmarks of the progression of clear cell to metastatic competency or aggressive disease. So as we were writing this paper, and even after it was published, I was beginning to have an existential crisis that I still have now, which is asking myself what changes in metabolite abundance actually reflected. So when I saw, for example, that citrate levels were accumulated in clear cell tumors relative to normal tissues, I didn't understand what the implication of that observation was for metabolic flux. And so I started asking myself maybe a more achievable question, which is whether changes in metabolite abundance were one-to-one, in a sense, with changes in gene expression. And if so, could we say something about metabolic flux? And the answer to that question is a resounding no. So if you compare, for example, at the pathway level, changes in metabolite abundance and changes in gene expression, tumor versus normal, you will see that in clear cell, almost every metabolic pathway downregulates the expression of genes composing that pathway. Whereas from the metabolomic perspective, metabolites are much more heterogeneous. And you can see that in detail on the right. This is central carbon metabolism, glycolysis, the PPP, and the TCA cycle. The ovals are metabolites, the rectangles are genes, and nearly all of the rectangles, nearly all of the genes are blue. They're almost all downregulated in tumors relative to normal tissues, but the metabolites show much more heterogeneous patterns. And so if you make that observation like I did, you might find yourself asking, well, how do I conclude anything about flux from that kind of data? And moreover, what can we learn, if not flux, from analysis of large-scale metabolomic data sets of primary tumor specimens? So that is the question that I'm going to try to walk you through in this talk in a variety of ways. And I'm going to give you a series of responses or answers that I've sort of come to over the arc of my research over the last decade. So maybe the simplest response, and the one that came fastest, was that we can, through analysis, essentially computational analysis of metabolomic data, sometimes discover new metabolic subtypes of diseases and generate hypotheses about their causes, about their etiology. This is really best typified by what happened right after this clear cell paper was published, which is that we noticed actually in the QC of that data that of our 140 tumors, there were two that were misclassified as clear cell, and on PCA analysis, they essentially were major outliers. And when we looked in detail under the microscope, we noticed that these really weren't clear cell tumors at all. They were sort of an emerging entity in the RCC field called clear cell papillary. So they had morphological features that resembled both clear cell and papillary renal cell carcinoma. And actually, the pathologist who identified CCPAP at first is a pathologist here at MSK, which is very fortuitous for us, because he helped us identify more CCPAP tumors that we could molecularly profile. So you might ask yourself, okay, well, what made these tumors outliers in the metabolomics data? And believe it or not, it was really one metabolite that was so overwhelmingly different that it drove these samples to be outliers. And that metabolite was sorbitol. So both in our original data set and then in a validation data set, we saw that sorbitol ended up being something like 30 to 100-fold higher in abundance in CCPAP tumors than in either clear cell tumors or normal tissues. So what is sorbitol? Well, it's a sugar alcohol, and it's produced essentially through an alternative metabolism of glucose. And the important thing to know about sorbitol is that in order to make it from glucose and then metabolize it, you essentially have to transfer an electron from NADPH to NADH. So if you know that, then you might come to generate a hypothesis about what's going on here. Because we know that kidney tumors are mitochondrially impaired. And if you see an accumulation of sorbitol, that might suggest that the subsequent downstream reaction may not be able to carry a lot of flux. And that reaction is an NAD-dependent reaction that makes NADH. So when I see NADH, I think of the mitochondrial electron transport chain and complex 1. And so I started hypothesizing that maybe there was something disrupted about complex 1, which is the primary source of NADH oxidation in cells, specifically in clear cell PAP, in CCPAP tumors, that maybe complex 1 was disrupted. So I was a young postdoc at the time, and I tried to learn how to call mtDNA mutations in tumors, which is actually how I met Scott Kennedy at first. So the thing is that, well, we tried to call mtDNA mutations from whole exome sequencing of a few clear cell papillary tumors, and we failed. And I learned that the reason that we were failing is that there was no coverage of the mitochondrial genome in these samples. And that was really puzzling because, well, mtDNA reads were abundant in all kinds of other whole exome sequencing data that we had from RCCs. And even in whole genome sequencing data, in some cases, the coverage was inadequate to really call variants. So it seemed as if maybe the expression or the copy number of mitochondrial DNA was low in clear cell papillary tumors. And that's what we actually went on to show. So from the perspective of either RNA sequencing, which is in the middle here, or protein expression on the right, looking at either mtDNA encoded protein expression or nuclear DNA encoded expression of a mitochondrially localized protein, CCPAP tumors really deplete themselves of mitochondrial mass and specifically of mtDNA and mtDNA encoded proteins. So it seems like clear cell papillary is actually defined by this very strange mitochondrial depletion phenotype that ultimately probably causes an accumulation of sorbitol. Okay, so what else can we learn from analysis of large-scale metabolomic data sets? Well, I would posit that we can actually learn something about the basic architecture of metabolism. That's not really a translational question, but it's really a question about basic biology. So what do I mean? Let me give you an example. So building on the original clear cell study, together with Ari Hakimi, my graduate students, Reese Tang, and I decided to conduct a follow-up study where we multi-regionally profiled clear cell tumors using metabolomics, RNA sequencing, and DNA sequencing. So for, I think, 31 tumors, we took anywhere from 3 to 5 regions in the tumor and around 2 or 3 regions in each normal kidney. And we did DNA, RNA-seq, and metabolomics. And the goal is really to assess intratumoral and intrapatient metabolic heterogeneity in these samples. So what we found on our analysis is that CCRCC and probably other kinds of tissues, they exhibit a very, I would say, almost universal pattern of intratumoral metabolic heterogeneity. So schematically, imagine that in a kidney tumor, you profile 2 regions and you see that one of them has higher citrate levels than the other. What you'll probably see is that in the citrate high region, there's high levels of cis-aconitate and low levels of fumarate. And if you repeat this whole exercise in a second kidney tumor or a third kidney tumor or a fourth, you'll see the same exact thing. So citrate high regions in a kidney tumor all have high levels of cis-aconitate and they have low levels of fumarate. And you can actually extend this analysis in an unsupervised way to the entire metabolome, essentially regressing out the baseline patient level metabolomic variation, and then doing principal components analysis to understand how metabolic fluctuations or metabolite fluctuations around the typical mean of a patient are correlated to each other. So if you do that kind of PCA analysis, you'll find that tumor normal regions are always separated by PC1, and I apologize, I think the blue and the red here are switched. So PC1 separates tumors from normals, and then PC2 is interesting because every patient seems to have PC2 high and low regions, but not necessarily PC2 high and low normal regions. PC2 basically segregates or discriminates between different regions of the tumor, but not of the normal. So, oh, that's weird. I lost it. Okay, so I have to tell you, I think I lost the slide. I have to tell you what PC2 actually corresponds to. So it turns out that if you look at in a tumor, PC2 high and low regions, you'll find that the PC2 low regions have high levels of a certain constellation of metabolites. They have high levels of cysteine, high levels of polyunsaturated fatty acids, high expression of OXPHOS genes and of a ROS signature, and also high abundance of certain antioxidants like vitamin E. So what is the biological process that connects all of these things? Well, we thought that they seem to be all associated with ferroptosis. So ferroptosis is essentially a cell death program where polyunsaturated fatty acids can undergo lipid peroxidation, and that can trigger iron-mediated cell death. And the cellular response to compensate or protect from ferroptosis is essentially mediated, at least in part, through the synthesis of glutathione, which is an antioxidant. So it seems like in every kidney tumor, you have regions that are very susceptible to ferroptosis because they have high levels of PUFAs. And to compensate for that, the tumor upregulates biosynthesis of glutathione and other antioxidants, essentially through upregulation of cysteine. So one other thing that we noticed, let me just see how this, yeah. So one other thing that we noticed in our analysis when we started modeling the covariation between the metabolome and the transcriptome is that the genes which seem to be associated with metabolite variation across tumor regions were strongly enriched for immune-associated genes. And in fact, what we noticed is that certain immune cell populations, especially myeloid populations, which are significant components of the CCRCC microenvironment, are strongly associated with something like 20% of the metabolome. So this nominated I would say, a pretty interesting hypothesis, which is that some of the intratumoral variation in metabolite levels that we're observing is actually due not to changes in tumor cell phenotypes, but actually to changes in the cellular composition of the microenvironment. Now, to corroborate that, we leveraged this internal database that my lab has sort of been building for a while, and that we have a preprint of, and that I encourage all of you to reach out to me if you want mine. We're happy to give you access to it, which is called the CAMP, the Cancer Atlas of Metabolic Profiles. So what is the CAMP? Essentially, it's a collection of mostly published paired metabolomics and transcriptomics data from different cancer types. So the camp has something like 1,000 samples of matched RNA metabolite measurements, primarily for tumors, but in some cases with adjacent normal tissues as well. So one thing that we can do in the camp is we can sort of repeat this analysis of looking for covariation between gene expression and metabolite levels. And we see the same sort of phenomenon that we saw in the clear cell data set. So I'm showing you a bit of a variation on that analysis where we've scored every single sample in the camp using an RNA signature of immune infiltration that's been previously published called immune score. And what you can see is that there are certain metabolites which are enriched in NAD-associated metabolites that strongly associate with just the gross infiltration of the tumor with immune cells. And now we've done this in a more granular way to try to essentially identify the key immune cell subtype or subpopulations that are really driving this effect. So I'm going to talk a little bit more about the camp in a minute. So the last thing I want to say here is that the idea that RNA levels and metabolite levels exhibit strong covariation is actually a bit of a trick that we can use to do something useful. So what can we do? Well, a year ago, my lab published a method in collaboration with Wes Tansey here, who's a machine learner, to basically address a prevalent problem in the metabolomics field, which is that when you do a metabolomics experiment and I do a metabolomics experiment, we often measure non-overlapping sets of metabolites. And if I want to know what my metabolite looks like in your data set, I can't. So we have a method that basically uses non-negative matrix factorization to impute unmeasured metabolites using essentially our data as a reference to learn on your data. So you can apply the same exact framework to impute metabolite levels from RNA data. And I'm showing you an example of us trying to do that in the TCGA by training a model to learn metabolite levels from RNA in our own in-house data of clear cell tumors. So what I'm showing you on the right on the x-axis is that in our tumors, every dot is a metabolite. And we've calculated the fold change essentially between tumors and normals in our data on the x-axis and in the imputed TCGA data on the y-axis. And you can see they're very consistent with each other. So we're actually imputing what we believe to be the ground truth tumor-associated changes in metabolite levels. And similarly, the changes between high and low stage tumors in our own ground truth data are exactly recapitulated in the TCGA data. So what can you do with this? Well, clear cell RCC has recently been the subject of numerous clinical trials with RNA biomarker analysis, where people have essentially tried to find transcriptomic biomarkers of response either in the experimental immunotherapy arms or the control, typically TKI or anti-VEGF arms. So we hypothesized that because a lot of the drugs that people are being treated with might perturb either the immune microenvironment, which can perturb metabolite pools, or directly perturb metabolism, that metabolite levels themselves might be associated with response to some of these therapies. So we used Mirth to basically train a machine learning model to learn RNA, to learn metabolite levels from our RNA data, and then applied it to 7 clinical trials in ccRCC. And what we found is that while there are no sort of reproducible biomarkers of response to immunotherapy in clear cell, there are actually reproducible metabolite biomarkers of response to anti-VEGF or TKI therapies in clear cell. And the sort of prototypical hit that we have is 1-methyl-imidazole acetate, which is essentially the downstream catabolite of histamine. So it seems as if patients who are more inflamed tend to have inferior responses. Tend to have superior responses to sunitinib treatment. And we think this is essentially a really promising method that one could use to impute metabolic biomarkers in lots of different settings where you have a reference data set to learn on. Okay. So my third response to the organizing question of the talk is that one thing that we can learn from large-scale analysis of the metabolome is something about like the conserved hallmarks of tumor metabolism biology that might transcend tumor lineage. And I was sort of showing you a little bit of that before with the CAMP data. And essentially all of this started when I realized that while as a young PI, I essentially had no money to make my own data, the cancer metabolism field had conveniently produced a lot of primary tumor metabolomics data for me that had been published, but not sort of collected and harmonized. So together with Augustin Luna on the right, featuring that dramatic black and white photo, I set out collecting and harmonizing basically 11 different studies of published tumor metabolomics data. So basically you made something like a pan-cancer atlas of the tumor metabolome. And when you do that, and then you compare tumors to normal tissues, you see something really cool, which is that there really are some metabolites which tend to recurrently accumulate or become depleted in cancers. The number one metabolite which accumulates in tumors is not anything to do with the Warburg effect. It's not lactate, it's actually kynurenine, which is the immunosuppressive metabolite derived from tryptophan. And I'll tell you more about that in a minute. One of the cool things about this data set is that we had associated clinical data with some, but not all of the studies. And so what we can do is sort of a meta-analysis looking for associations between metabolites and tumor grade or tumor stage that transcended cancer lineage. So in the same way that I was telling you that clear cell tumors seem to be metabolically reprogrammed in high stage disease, do we see something similar across all cancer types in the same metabolites? And indeed we do. So for example, kynurenine levels seem to rise in high-grade tumors of a number of cancer types. And what I would say is more interesting is that if you look at, for example, spermidine on the right, it's not just that spermidine levels in the tumor changed with grade. It's actually that the spermidine levels in the normal tissue next to high-grade tumors tend to be higher. For example, in kidney tumors, they tend to be higher than spermidine levels in normal tissue next to a low-grade tumor. So it's almost as if there's a field effect going on in some of these diseases. So that early study led us to build the camp. And really the motivation for the camp was not to necessarily learn something about tumor metabolism, but basically to use free data in the cancer metabolism field to learn something about the architecture of how genes and metabolites are co-regulated. And to do this, we basically did something like a concordant, well, a meta-analysis with the goal of trying to find gene metabolite covariation that transcended different data sets in the camp. So what did we find? Well, we found a lot of hits. So something like 22,000 gene metabolite pairs that seem to be statistically significantly covarying across our giant data set. But when you actually dug into what those 22,000 hits constituted, there were two really clear patterns that I'm going to share with you. So the first is that there are a very small number of very strong hits. For example, IDO1 in kynurenine, GDA in guanine, and CD38 in NMN. And the interesting thing about these very strong hits is that they are proximal interactions. So IDO1 acts not quite directly, but essentially directly on kynurenine. Guanine is metabolized by GDA, and I think NMN is either produced by CD38 or is sort of one step away from CD38. So these really strong interactions seem to represent almost like mechanistic enzyme substrate interactions. But if you look at panel B here, what you'll find is that of our statistically significant interactions, only like 2% of them are examples of these proximal pairs. So 98% of our hits are not between genes and metabolites that are obviously related to each other. So what is the other 98%? Well, actually, before I tell you, let me just say that one of the interesting things is that we noticed that, well, IDO1 has been sort of, I think, well established to regulate the abundance of kynurenine. And in fact, there are drugs that have failed trials in patients whose goal is to inhibit IDO1 and thereby decrease the abundance of immunosuppressive kynurenine in the microenvironment. But the interesting thing about IDO1 is that it's actually a significant hit, not just for kynurenine, but also for tryptophan. And that's really surprising because tryptophan is a proteinogenic amino acid that is involved in, the biosynthesis of essentially every protein. So it was puzzling to us, can IDO1, this sort of strange enzyme, really regulate the pool size of tryptophan? So we did that experiment. We knocked out IDO1. We confirmed that knocking out IDO1 decreases kynurenine levels. And interestingly, knockout of IDO1 actually increases tryptophan levels as well. So it seems like the abundance of this proteinogenic amino acid is really, at least in part, controlled by the expression of IDO1. Man, I thought I was going to tell you guys about something else, and yet I didn't. So let me give you the answer to what the rest of this data is explained by. So the other 98% of our hits here, they are explained interestingly by the cellular composition of the tumor samples that we profiled. It turns out, as I sort of mentioned before, that the infiltration of the specimen by immune cells seems to strongly reorganize. And it turns out that there's something like 10 metabolites that account for 20% of all of our hits. So out of the 300 metabolites that we consider, 10 of them account for 20% of our hits. And the genes that those 10 metabolites associated with are all expressed specifically on immune cells. So what that suggests is that immune cells seem to be poised at potentially a different metabolic state with different characteristic concentrations of those metabolites, which predominantly are NAD metabolites. And so when immune cells are infiltrating the tumor to a large or a small extent, that can drive almost artifactual correlations between metabolite levels and gene expression. Okay, so the last thing I want to talk about is, well, the last thing that I think I learned about metabolomic from metabolomic data analysis is that it can often point the way to where we should look in the genome to find interesting phenomena. So this is really typified by the clear cell papillary story that I told you earlier. So once I learned how to call mtDNA copy number from the clear cell papillary work, I thought, why not? Let's do it in the TCGA. And as it turns out, the TCGA sort of oddly ignored mitochondrial derived reads when doing their analysis. And the rationale there was actually that they were doing whole exome sequencing. And so they were intentionally trying to deplete mtDNA. And so I guess they weren't comfortable calling variants or copy number from those reads, even though their depletion wasn't very effective and those reads were still abundant in the data. So I went back and recaptured those reads from whole exome sequencing. And what I discovered is that when you compare tumors to matched adjacent normal tissue, about half of solid tumor types seem to be depleted of mtDNA copy number relative to normal tissue. And I found that to be really exciting. And I realized that maybe there's a bit more to learn about the mitochondrial genome than had sort of been disclosed to that point. And what got me really excited about working on mtDNA was that from my perspective, I thought, well, mitochondria and the proteins encoded by mtDNA essentially participate in the canonical metabolic decision that every cell makes, which is whether to respire or to ferment. So what I mean is, if you look at a well-differentiated, terminally differentiated tissue, in the presence of oxygen, that tissue, if it's signaled appropriately, will take up glucose and metabolize it to pyruvate that will eventually enter the TCA cycle and then undergo oxidative phosphorylation in the electron transport chain and make a bunch of ATP. And it's only in the absence of oxygen that that tissue will metabolize glucose less efficiently to lactate. So what happens in cancers and in many proliferating tissues, like let's say activated T cells, is that whether or not oxygen is around, glucose will be preferentially routed to lactate, and only a subset of glucose will be catabolized in the mitochondria. And some people call this Warburg effect or aerobic glycolysis. And there is essentially a century of hyped and controversial literature on this topic. So I also became really interested in studying mitochondrial DNA because, well, I came from, my PhD is really in bacterial metabolism, and the mitochondrial genome is basically a really cool bacterial genome inside of each one of your cells. So it's the highly compact genome. Some of the genes don't even have stop codons. Those stop codons are actually created by polyadenylation. It encodes 13 protein coding genes. It's present in hundreds of thousands of copies in each cell. It has its own translational machinery and its own genetic code. So it has tRNAs and ribosomal RNAs. And those can become mutated in a way that you would never see a tRNA become mutated in the nuclear genome. And actually, mtDNA mutations are essentially the most common cause of inborn errors of metabolism in the germline. And really, most provocatively, it turns out that, and this was already established when I started, 50% of all tumors have a somatic mtDNA mutation. So the question was really, was it Titan? Just are these all passenger mutations? Was it P53? Or was it somewhere in the middle? And when I started working on this, I had just started a pre-tenure track faculty job. I didn't have the money in my startup package to do sequencing. And I was competing essentially against the PCOG and ICGC whole genome teams that were well-staffed and well-funded and had whole genome data on the mitochondrial genome. So to compete against them, together with Payam Gramaj, who's an excellent PI and essentially my best friend scientifically at the Beatson, and our graduate student, Alex, we realized that we could use the exome sequencing data that I'd used to call mtDNA copy number in cells in the TCGA in tumors, to also call mtDNA mutations. And the thing is that if you actually go to the TCGA and you try to do this, depending on the exact way that you call mtDNA mutations, without doing anything really, you can double the statistical power that is available to the whole genome PCOG cohort. So that's what we did. We built a pipeline to use off-target reads to call mtDNA mutations. And the reason you needed a pipeline was basically that in many cases, only parts of the genome were covered in any given sample. And so we had to sort of statistically think about how to aggregate piecemeal signals across samples. We established that our pipeline works quite robustly. And then we went about our work. So if you calculate either the truncating mutation burden of mtDNA or just the non-synonymous burden of mutations to mtDNA in tumors, it turns out the mutations to these genes are essentially as common as mutations to the most common tumor suppressors that we study every day in cancer. So from the perspective of truncating mutations, ND4 basically suffers as many mutations, as many indels or nonsense mutations as everything except P53, P10, and CDKN2A, which are the subject of probably millions of research articles in PubMed. And similarly, complex 3 in mtDNA is actually more mutagenized than PIK3CA and NRAS and IDH1. So these are real deal mutations. And the question is, do they have any function? So, you know, the prior literature to this point in the mtDNA cancer literature had focused on calculating DNDS, which was a measure of evolutionary positive or negative selection. And we took a different approach, which was basically to look for differential selection for mtDNA mutations as a function of two things: the respiratory complex that they impaired and the kind of mutation that was produced. So we know that the electron transport chain in mtDNA, well, it is basically what the proteins in mtDNA, they are subunits of the electron transport chain in ATP synthase. And we know that when you perturb different components of the electron transport chain, either pharmacologically or genetically, because many of those components are nuclear DNA encoded and can be CRISPRed or whatever, depending on the complex that you perturb, you get different phenotypes. And so we kind of hypothesized that the evolutionary selection for or against mutations should depend on complex. And that's exactly what you see. So really pathogenic truncating mutations to mtDNA have a strong preference for complex 1 and are strongly selected against in complex 5. And that's not something that you see in silent mutations, where the burden is essentially uniform across the entire genome. So more interestingly, when you look at truncating mutations, not only are they preferentially in complex 1, but they arise in hotspots, which are sort of like the calling card for selection in cancer genomics. So we identified something like 6 hotspots in the mitochondrial genome, all in complex 1, all at very specific homopolymers, not just all homopolymers, and all in specific cancer types. So it turns out that tumors of the kidney, of the colon, and of the thyroid were really the subjects of these mutations. So even though kidney, thyroid, and colon seem to bear the biggest burden of these mutations, we found essentially at least one instance of a truncating mutation in all cancer types. In fact, something like 1 out of every 10 tumors, regardless of cancer type, has one of these mutations. And using matched RNA sequencing data from the TCGA, we can compare the phenotypes of tumor samples that had truncating mutations to tumors that were wild type for mtDNA. And interestingly, what we found is that the presence of a truncating mutation was sort of associated with a lineage agnostic transcriptional phenotype. So regardless of whether it happened in the kidney or the colon or the breast or the lung, mtDNA mutant tumors or tumors with truncating mtDNA mutations seem to upregulate OXPHOS genes and downregulate genes associated with the innate immune response. And that suggested that they were functional, at least to some extent. And so the question for us then became, well, I mean, these genes are sort of quasi-essential. Certainly, malignant cells will not do very well if they have a completely impaired complex 1. And so we reasoned that actually the basis for cellular tolerance of these mutations is heteroplasmy. So what is heteroplasmy? Well, I told you before that every cell has something like 1,000 copies of mtDNA. And when a mutation arises, it typically only affects a fraction of the total pool of mtDNAs in a cell. And that fraction is called the heteroplasmy. So in sequencing data, it would be like looking at the allele frequency, although every cell in a specimen will probably have a different heteroplasmy. So our hypothesis was essentially that, by arising heteroplasmically, cells could preserve the wild type pool of mtDNA, but by increasing the dosage of the mutant pool just a little bit, they can promote, let's say, pro-oncogenic functions. And if that's the case, then you should see selection. In cases where you think there's positive selection going on, you should see selection for higher heteroplasmies. And that's actually exactly what you see in complex 1, but not in any of the other complexes. So if you compare the distribution of heteroplasmies in truncating and silent mutations by complex, you see that only complex 1 shows an increase in heteroplasmy over silent mutations. which sort of supports but doesn't really prove that they're under positive selection. So I just want to touch a little bit in like the last few minutes on what else can happen in the mitochondrial genome that I think is really fantastic. So there is a rare, but actually quite nasty and malignant form of thyroid cancer called Hürthle cell carcinoma. So this is a disease that when you look at it under a microscope, the cells look like they're swollen with dysfunctional mitochondria. It's oncocytic. And so for a long time, people have presumed and actually proved that these tumors have mtDNA mutations in them. And in compensation to those mtDNA mutations, the tumor seems to upregulate mitochondrial biogenesis, or alternatively, just stinks at clearing dysfunctional mitochondria. So together with Tim Chan and Ian Ganley here at MSK a few years ago, we sequenced something like 50 or 60 Hürthle cell carcinomas. And our study came out exactly at the same time as a study from Vamsi Mutha's group from Boston. And what we found was, interestingly, that yes, of course, these tumors had truncating mtDNA mutations, almost all in complex 1. But more interestingly, they seem to have a hotspot mutation in a mitochondrial tRNA. So this isn't totally surprising. In fact, I think the most common mtDNA mutation that causes disease when inherited in the germline is a tRNA mutation at position 3243 in the genome, and it causes a disease called MELAS, which stands for something like mitochondrial encephalopathy and lactic acidosis. It's a really nasty disease. And you might be asking yourself, well, okay, well, how do cells tolerate a tRNA mutation? Well, remember that these tRNAs are only essential for the translation of mitochondrial proteins. So even complete dysfunction of the tRNA would only affect mitochondrial translation. So what we found is that the mutations in Hürthle cell affected not 3243, but actually 3244. And interestingly, in 2005, there was a group that characterized what would happen in cells that had exactly the mutation we were seeing at 3244. And essentially what it does is it disrupts post-transcriptional modification of the wobble codon in the tRNA that gets modified during folding and processing. So again, this is actually an example of how the genetics ultimately informed a metabolomic experiment. What we ended up doing is actually metabolomically profiling these Hürthle cell carcinomas. And you can look at this data in the camp if you like. So these tumors have, again, a really remarkable metabolomic phenotype that's almost, I would say in some ways, it's kin to clear cell tumors, but in a really puzzling way. So remember, these tumors are like homoplasmically mtDNA mutated. I mean, they have almost no wild type mitochondria in them. So presumably they at least cannot conduct complex 1, although they can probably do other stuff. So what we see is that these tumors seem to be almost completely depleted of their pools of citrate and cisaconitate. And we don't really know the functional significance of that. We don't know how it's produced. We have some cell line data where we've done tracing, where it seems like even in the background of these mutations, glucose-derived carbon can still enter the TCA cycle and make citrate. But I guess, I would draw as a point of contrast, clear cell renal cell carcinoma, which has been proven both in cell lines and in patients to conduct aerobic glycolysis, to preferentially shunt glucose to lactate. And there, when you know that glucose-derived carbons are not entering the TCA cycle, you see lots of citrate, much more in tumors than in normal tissue. This is presumably glutamine-derived. So it seems like what's happening in Hürthle cell, in contrast to clear cell, is that presumably glutamine-derived carbons are not replenishing the citrate pool. Okay, so I've taken you on a whirlwind tour and have probably confused you. So let me summarize a little bit. I hope I've convinced you that metabolite alterations are really pervasive in tumorigenesis and the progression to aggressive disease. But I hope I've also convinced you that you should be at least a little bit disillusioned with the measurement of metabolite levels, in that they are not good proxies for flux, just like gene expression is not a good proxy for flux. But just because they are not does not mean that they cannot offer you insights. So you can learn about tumor etiology, or say the structure of your cohort from that kind of data. You can understand the physiological factors driving intratumoral variation. You can learn about basic metabolism from looking at metabolite gene covariation or even metabolite covariation. And the things that you learn, at least from our approaches, transcend the tissue of origin. And finally, I mean, the study of metabolite levels is a really good way, both in the cancer field, but also in like the inborn error field, of finding interesting metabolite-associated genetic loci. And finally, I hope I've sort of convinced you that somatic alterations of mtDNA are overlooked, and that in very specific contexts, in kidney cancers and thyroid cancers and colon cancers, they can be under really significant and recurrent selection. And with that, I'll stop and thank the people who did the key work in the group. And our funding sources, I'll emphasize that my lab is hiring. So if anybody is interested in talking about that, please reach out to me. And now I'm obligated to show you this slide so you can go home and eat dinner. Thanks very much.
Thanks, Ed. That was a whirlwind, and that was fantastic. That's a lot of work, and really, really innovative for you to sort of use those off-target reads. I mean, that was great. So I guess I'll start with the questions then. So everyone, you can type a question into comments again if you'd like, or just speak up. Either one should be fine. I'm monitoring the chat. Well, Ed, one of the things I was sort of curious about was when you talked about the spermidine in the peripheral cells and the sort of environment of the stroma around the tumor, that you can detect that sort of thing. One of the things that's sort of come up in the last 5 years or so in tumor biology is the sort of actual like microbes, that there's actual bacteria and fungus in some tumors, and even in kidney tumors, some of the papillary ones have abscesses. Does that how do you think that, have you considered that? Do you think that plays into anything? Is that something that, is it too infrequent, or is it too new, or is it?
It's a good question. I have to say, sorry, I'm going back to it. Not that it really matters, but it's interesting that you raised that, because I haven't thought about it in this context, but actually when you look in our pan-cancer data where we analyze gene metabolite covariation, several of the metabolites that seem to be strongly covariant with genes outside of NAD metabolism, they're actually non-human endogenous metabolites. They are like bacterially derived. And
Fascinating.
I always kind of wondered about it. I've asked some ID doctors here who kind of suggested that, when patients are under therapy, they can have a really disrupted microbiome, and you can have those metabolites seep into the circulation. I don't know. I really never made much of it.
It's just been fascinating. So one of my buddies was looking at 80 patient data, and then they looked at the control brains, and they found out that people threw out a bunch of reads from the control brains that were bacterial.
Right.
And then people thought, well, oh, that was just bacterial contamination. But now, like 5 studies later, there's a brain microbiome. There might be microbiomes normally all over us in places that we never thought, that maybe just because we can't culture it, we didn't see it. But boy,
yeah,
the evidence seems to be there in the seq data.
Yeah, it's a good idea. And I don't know. I mean, yeah, my whole career is basically repurposing data. So I don't think it would be very hard to go back, for
That's
example,
what
to
Chris
the
Link
RNA
did.
data. Yeah.
Yeah, and he was just sort of a novice at RNA-seq. So he's like, why is this getting thrown out?
So are the bacterial reads polyadenylated? Like, do they get captured in poly-AC?
I don't know all of the details. I think it was just like a random hexamer thing, right? because they shouldn't be, but I don't know. I don't know all of the details. I just sort of from a fanboy perspective that this is sort of an emerging thing that, in 2013, these Brazilian guys were sort of cast into PLOS One because that couldn't be right. And then, 5 years later, somebody at Harvard finds it and then it's correct. So,
Yeah, that's it's a good thought. I had never thought to do it, but as I mentioned, it might explain some of our hits for sure.
It seems to be a thing. So there's a Science article on it, and in the last 5 years, people seem to be accepting that there's like the tumor cells seem to be having some sort of interactions with the microbes, and that there might be, a little bit of a back and forth, like, hey, would you guys like to break off and form your own state? we're cool with that.
Yeah.
But yeah, it's just your work's really, really clever and innovative, and I really do like that you have a new perspective that you're bringing to it, just because you learn new things. So I love this. I think that this is really, really important. It'll really help out with understanding. Just for me, I always get into the heterogeneity of things too, but just, some of the differences between tumors, and this is all really, really fascinating. And I mean, so what do you think that this provides for therapeutical insight?
That's a good question.
For your translation sentence in your grant application.
Yeah. I have to, to be totally honest, I'm skeptical about the therapeutic insights that can be gained from it. I mean, certainly I showed you that I think we can get metabolite biomarkers of response to certain therapies. And I think there is some
prognostic biomarker value, like,
yeah, well, only because some most of the time pool size doesn't matter. Most of the time metabolite abundance doesn't matter, but there are immunosuppressive metabolites that are the targets of therapies. So kynurenine, adenosine, sometimes NAD, so there is potentially some rationale there. And if you can really train an RNA model to impute metabolite levels, that's really useful because
absolutely,
you don't need fresh frozen tissue, essentially. But then the other way that I think is, maybe a better answer for you is that I think that potentially these kinds of approaches can be used to identify metabolic changes that can ultimately be leveraged in a non-invasive way to monitor something like therapeutic response. So if we know that, let's say from pre and post therapy RNA sequencing data, that there's a certain gene signature that is associated with a metabolite that changes in response to a therapy, you can develop a PET tracer, for example, to monitor that. And that's really a problem in kidney cancer. There are really no non-invasive approaches to monitor a response. So it would fill a significant clinical need. Yeah.
That's awesome. So you could develop biomarkers that would allow clinicians to get maybe some cell-free DNA or RNA from sloughed off kidney cells, perhaps.
Yeah, potentially. You would have to train a new model, but yeah.
Yeah, so that's, just imagining the translation of the clinic so that they can know for this kidney patient that this patient's responding now, but this other patient isn't. So with that information, they could intervene and change therapies.
Mm-hmm.
That's exactly. To me, this work, it seems it was a layup for translation, I thought, because of the biomarkers, right?
Yeah.
Biomarkers are really, really, really valuable, and machine learning can identify biomarkers that people didn't even think about just because it's the machine. So it's like your approach. I'm really hopeful for what this could do for personalized medicine and for helping people out. This is really great. Everybody in Path 520, remember to scan the QR code and submit your evaluation. And does anyone else, I know I've been very chatty. Does anyone else have any other questions for Ed?
I think that there's a question in the chat. I can read it out. So
yeah, that would be great. I did not see
questions from.
Oh, I see it. I see it. Here we go.
Yeah. So the first one is, do metabolic and metabolite alterations scale with the extent of heteroplasmy? So the answer is unknown, but I will tell you that there are now genetic tools to introduce novel mtDNA alleles. They're called DDCBs. So together with Payam Gamage, we've been working on that. And the interesting thing is that when you introduce one of these novel mtDNA alleles that, let's say, mimics the one that you see recurrently in human tumors, they do produce subtle metabolomic changes. They're not grand, but those subtle metabolomic changes can have significant effects on the local immune microenvironment. That's sort of the phenomenon that we see, that essentially there's small changes in O2 consumption or no changes at all. There's some changes in metabolite abundance, but those changes are really sufficient in a dosage-dependent way to change the local microenvironment. And then the second question is, are there clear signals you can use to identify stromal cell type specific contributions to metabolic profiling? I'm not sure if I understand it, but if you're asking if stromal cells have a particular metabolite signature, I can say that it's probably going to be tissue specific, but in kidney, yes. And if you want, I can send you a list. I actually don't remember offhand which metabolites associate with highly stromally, like infiltrated or stromally abundant kidney tumors, but we just have that tabulated, and I'd be happy to share it.
Cool. Ray, did that answer your question? I hope.
Let me see if I can unmute. Can you hear me?
Yeah, we can hear you, Ray.
Oh, great. Yeah, thanks. Thanks. I really enjoyed the talk because this is an area I've thought about for a long time. The second of the questions, I'm thinking of genomic parallels here, where people have been able to take like RNA expression data and deconvolute it to identify both the type and the contribution of different, say, immune or inflammatory cells to a tumor sample. And a lot of what you presented, I'm assuming, was in the context of tumor sample profiling.
Well, yes. Yes, it is. But remember, the tumor samples are cellularly heterogeneous. Yeah.
Exactly. Yeah. Yeah. Okay.
So, so let me see if I understand. So you're sort of asking, in a way, if there are, for example, metabolites that are specifically at higher or lower concentration in stromal cells. Is that right?
Well, I was wondering if you could identify, say, a 20% contamination of a particular type of lymphoid or inflammatory cell.
I think we could go halfway to it in that, for example, if you told me how much, let's say you measured 10 tumors, and in those 10 tumors, you measured the amount of quinolinate, which is an NAD-derived metabolite. Whatever the ranked ordering is of quinolinate, that's the ranked ordering of how immune infiltrated, just grossly, those tumors.
Okay.
Yeah.
Yeah.
In terms of specific cell populations, you can almost do it. So for mast cells, if you tell me how much histamine each of those 10 samples has, that's how many mast cells they have in a non-parametric way. It's not quantitative, but semi-quantitative. Yeah.
Yeah, okay. And I know we're running late. I'll just ask one last quickie here. It's a technical question Alex, I thought, would answer, which was, how close are we to doing spatial profiling of the metabolome?
Oh, it's done. I think spatial profiling of the metabolome is like the oldest spatial technology. It's called MALDI.
Yeah.
Well, okay, the oldest spatial technology is imaging. So, you know.
Yeah, okay.
But in terms of molecular spatial technologies, yeah, MALDI has been around for a while.
Yeah.
The amazing development in MALDI, I would say, in the last 2 years is that they paired it with isotope tracing. So there is, I was talking about it before, that there are 2 papers that came out in the last year or two, one from Josh Rabinowitz and Sean Davidson, and the other one from a Dutch or Belgian group, where they've basically infused a mouse with labeled glucose and cut out the kidney and done MALDI. So spatial metabolomics, but they can resolve the different isotopes. So in vivo, you can see isotope labeling patterns that can help you sort of, infer flux ratios. Yeah. Yeah,
cool. I'll go back and look for those. That's a great tip. Thank you again.
Yeah, of course.
Well, I hope everyone would like to give Ed a virtual round of applause. Ed, that was a great talk.